Hi, my name is Elliot, and I have done research this summer at the University of Iowa, the Department of Internal Medicine. What we worked on was trying to determine the role of the gene GON4L in B and T cell lymphopoiesis. We determined that the GON4L was actually necessary for T cells to proliferate normally when they are already been matured, and we determined that by proliferation assays using radioactive markers. Then we determined that these T cells from our mutant that lacks GON4L do not express a protein called STAT5 as to the level that's necessary in normal T cells to have the normal proliferative response. We used RT-PCR to determine that SOX proteins were misexpressed in T cells, which provide a possible mechanism for the lower levels of STAT5 activation. We also continued our research in B cells and determined that there wasn't a significant difference in STAT5 activation in B cells. Continuing on, we tried to examine functional interactions between GON4L and transcription factors such as STAT5, and found that through co-immunoprecipitation assays that GON4L interacts with STAT5 either directly or indirectly. And this, we want to continue this research by looking into more interactions with other known transcription factors using transfections. This research is important because it tells us about the developmental processes required for normal immune cell development, which is useful in understanding what might go wrong when a lymphoma occurs or another cancer. Our Western blots for GON4L demonstrate that our mutant does not express normal levels of GON4L in their T cell population. Our proliferation assays show that the wild type proliferates normally in response to IL-2 and TCR signaling, but the Justy mutant does not. And then our Western blots for activated STAT5 show that the Justy mutant has decreased levels of activated STAT5. And then our FACS experiments show that this is not true in the B cells, that STAT5 activation is actually lower. And finally, the Western blot for STAT5, while immunoprecipitating for GON4L, shows that there's a direct interaction between STAT5 and GON4L.